Hi, this is Jeffrey Townsend at Yale University, and I'm here to tell you about a maximum likelihood method for clustering, which we've just published in PLOS Computational Biology. Maximum likelihood model averaging to profile clustering of site types across discrete linear sequences. A major computational challenge in computational biology is the detection and description of clusters of specified site types, such as polymorphic or substituted sites within DNA or protein sequences. Progress has been stymied by a lack of suitable methods to detect clusters and to estimate the extent of clustering in discrete linear sequences, particularly when there's no a priori specification of cluster size or cluster count. Here we present an algorithm that addresses this challenge, demonstrate its effectiveness with simulated data, and apply it to a celebrated example of genetic polymorphism data. To apply MACML to locate clusters of site types, your data must first be transformed into a vector of zeros and ones. For example, zeros might denote variant sites, and ones might represent polymorphic sites. We derive a null model assuming no clustering, and an alternate model incorporating heterogeneity of variant site frequency. The alternate model incorporates a tripartite divide and conquer strategy that captures diverse patterns of heterogeneity, delineates cluster boundaries, and also yields a profile of the level of clustering associated with each site. Here's how the procedure flows. You input your vector of zeros and ones. Indices I and j are assigned to the start and end site of the sequence. All possible single clusters are evaluated. If the most likely cluster is statistically significant, then each region, left of the cluster itself, or right of the cluster, is independently evaluated for subclustering. This subclustering is iterated until all potential subclusters are not significant. To validate our approach, we simulated data for which we know the underlying clustering. We looked at two metrics of success: the power to detect the true cluster and the Kullback-Leibler divergence, which, for our purposes here, you can think of as a metric of how far off our estimated clustering description is from the true underlying clustering. We compared our method to the best extant method for detection of clustering in discrete linear sequences, Tang and Lewontin's empirical cumulative distribution function, or ECDF. Here's the blow-by-blow. Examining the percentage of variant sites within the cluster across panels for 10 to 200 variant sites, MACML, in the circles, exhibits greater power to detect clusters than ECDF, in the squares, for both hotspots, in the upper panels, and cold spots, in the lower panels. Similarly, the KL divergence between estimated and actual clustering is near zero for MACML, but diverges significantly from zero for ECDF. Examining from extreme cold spots on the left to extreme hot spots on the right, both on top, MACML exhibits greater power than ECDF. Both the accuracy and precision of estimation, shown on the bottom, of cluster location is better with MACML, whose divergence is near zero, rather than much larger than that with ECDF. The power of MACML is greater than that of ECDF across sequence lengths, and the accuracy and precision of MACML is better across sequence lengths. MACML also detects multiple independent clusters better than does ECDF, to an extent that depends on the number of variant sites. Having shown the relative power, precision, and accuracy of MACML compared to ECDF, we have just one more thing to measure. We can compare the false positive rate of ECDF to the corresponding overparameterization rate of MACML using the three information criteria implemented: the AIC, AICC, and BIC. Examining these rates across 10 to 200 variant sites, we see the unsettling result that the rates are extremely high, nearly 100% in the case of AIC and AICC, and 20 to 70% in the case of the more conservative BIC. These rates appear tragically high. Is this the demise of this promising approach? In fact,
Is this the demise of ECDF and MacML, and the whole enterprise of discovering clustering in discrete linear sequences? How can we rescue ourselves from this graveyard of ideas?
The reason the false positive rates are so high is that there are so many possible clusterings, so many possible models for any given sequence. With so many possible models, it becomes extraordinarily likely that the most powerful method will identify one model as significant even when the clustering is produced by a process with no underlying correlations. What one needs to do is to incorporate all possible or all likely models, not just the most likely model, so that the null model is incorporated into the estimates themselves.
Because MacML's cluster recognition is based on information criteria, all likely models may be averaged to yield composite estimates that account for the uncertainty about the correct model. By averaging across models, cluster borders are appropriately softened, and a more realistic description of the heterogeneity underlying your discrete linear sequence is produced. Take the example of the ADH gene of Drosophila, a classic exemplar of genetic polymorphism. In panel A, using the BIC, MacML clearly picks up the well-known central cold spot of polymorphism and more. Using model averaging, this rigid portrayal of heterogeneity becomes much more refined, and the relative Akaike weights even facilitate the calculation of confidence intervals. Using the more permissive AIC, more detail of clustering may be observed, and model averaging reveals an even richer profile of polymorphism. So the model averaging methodology allows us to overcome the natural and extensive uncertainty in cluster borders, to incorporate the null hypothesis into our estimations of variability, and to provide a realistic profile of sequence heterogeneity and clustering, one founded on the principles of probability theory that is not dependent upon assumptions of window size or the number of clusters expected. MacML is freely available for download from the Townsend Lab website. We hope that the profiles of clustering and constraint that it provides will be of broad utility for computational analyses or visualizations of heterogeneity in discrete linear sequences, an enterprise of rapidly increasing importance given the diminishing costs of nucleic acid sequencing. So what music should we have here? Maybe we'll put on the Who's Magic Bus.